the United States disease labs made dangerous pathogen transport errors. The United States government infectious disease labs mishandled dangerous pathogens 5 times in the last decade, according to a health agency report. This year alone, workers mishandled samples of anthrax and the highly infectious H5N1 avian flu. In response, the Centers for Disease Control and Prevention (CDC) has closed the 2 labs involved. The agency has also temporarily barred high-security labs from transporting dangerous pathogens. There have been no reported infections from previous cases, and no one potentially exposed to anthrax has shown signs of illness, CDC officials said. These events should never have happened, CDC Director Tom Frieden told reporters on Friday. "I'm disappointed, and frankly I'm angry about it," he said, adding later he was astonished that this could have happened here. The incidents were listed in a report on a potential anthrax exposure in June, which occurred when researchers in a high-level biosecurity laboratory failed to follow proper procedures and did not deactivate the bacteria. The samples were then moved to a lower security lab in the agency's Atlanta campus. This is not the first time an event of this nature has occurred at CDC, nor the first time it occurred from the Bioterror Response Laboratory, the report said. The CDC only recently learned of a separate incident in May in which a sample of the avian flu was cross-contaminated with a highly pathogenic version of the virus and then shipped to an Agriculture Department laboratory. The Influenza Lab and the Bioterror Response Laboratory have been temporarily closed in response. The other incidents reported by the CDC, in 2006, the CDC's Bioterror Lab transferred anthrax DNA to outside labs, believing the sample had been deactivated. Also in 2006, samples from a different CDC lab were found to contain live botulism bacteria. Researchers sent an infectious strain of the bacterium Brucella to outside laboratories as early as 2001 because they mistook it for a vaccine version. Newly available test methods in 2009 confirmed it was not. The revelations come just days after the United States officials announced long-forgotten vials of smallpox left in a cardboard box had been discovered by a government scientist at a research center near Washington. The virus was located in 6 freeze-dried and sealed vials, according to the CDC. On Friday, the CDC announced that at least 2 of the vials contained the live virus but that no infections had been reported as a result of the incident. It is believed to be the first time unaccounted for smallpox was been discovered in the United States. The disease was officially declared eradicated in the 1980s.